Hello, my name is Kurt Moody, and I'm a professor of biology and environmental science at Columbia College in Columbia, South Carolina. Today I'm going to show you a model I developed to teach the double helix structure of the DNA molecule. It's a model that I've used with my students to teach about how the form of this molecule influences its function in lots of different biological processes. This type of modeling has been used to teach about DNA ever since we learned the structure. In fact, this isn't even the first DNA teaching model that I developed. A few years back, I made one out of paper, and if you're interested in that, it's currently available on the Howard Hughes Medical Institute Biointeractive site, which is a great teaching resource I highly recommend. Anyway, the model that I'm showing you today is actually built out of Lego blocks, which isn't really that unique. Many folks have built double helix structures out of these blocks. And if you do a search for Lego DNA, you'll note an amazing variety of educational and artistic efforts. I take this as evidence of the kind of enthusiasm that using Legos in an educational context can generate. This particular model, however, has some truly unique characteristics that make it much more valuable for teaching or demonstrating. So to start things off, I want to tell you what those characteristics are up front so you can decide if you want to watch another video on how to actually put the model together. So first up, I want you to understand that this is a completely freestanding model. There's no tape, there's no glue, there's no rod up the middle, just Lego pieces fitting together the way they always do in Lego structures. And as you can see, it is in the form of two helical strands twisted around each other, forming the DNA double helix. Each strand is built from a series of nucleotide subunits, and each nucleotide features a phosphate group, those are the red bricks, and a deoxyribose sugar, those are the blue bricks, and a nitrogenous base. Now, if you know about DNA, then you know that the phosphates and sugars are all the same, but the nitrogenous bases come in 4 different types. This model represents those types with bricks of 2 different sizes and 2 different colors. The sizes represent the difference between purines and pyrimidines, and the colors show the complementary pairs. So the nucleotides with large green bricks are adenines, the ones with small green bricks are thymines, the ones with large yellow bricks are guanines, and the ones with small yellow bricks are cytosines. So this model is showing nucleotide subunits, each with their component parts. And these nucleotides can be put together separately ahead of time, so that the builder is modeling the process of nucleotide synthesis separately from DNA synthesis, just like it is in a cell. It's also important to point out that the helical structure of this model is a product of nucleotide geometry. We're not building a ladder here and then twisting it into the double helix. Now, this is already an improvement over most DNA teaching models because they're often built around a stick that won't allow nucleotides to be removed, or sometimes they're built from things like a sugar phosphate backbone or a series of base pairs that can't be separated. But these strands are completely independent. In fact, they can be built separately. Or if the double helix is built, the strands can be shown separately at the upper edge. This is so important if the student is ever going to consider things like DNA replication or repair or transcription or PCR, which all require strands that have their own independent structure. All right, now here comes my favorite part. If you look carefully at the model, you will see that one of the strands is right side up and one of them is upside down. This models the anti-parallel structure of the double helix. Very few DNA models actually address this pattern, even the ones that are designed for teaching. And to my knowledge, none of the Lego models do it, so that's why I get a little bit excited. Also, take a look at how the anti-parallel strands produce 2 different types of grooves on the outside of the molecule. A narrower minor groove where the studs are facing each other, and a wider major groove where they're facing in opposite directions. These alternating areas play really important roles in the way that DNA interacts with lots of other types of molecules. Another amazing thing about this model is that it makes a complete turn in a little over 10 base pairs, which is right on target for DNA. And even the width to length ratio is about 2 to 3 for one turn, which is super close to the actual molecule. So just looking at it gives a good appreciation of relative scale. And then finally, for some bonus points, I want you to know that if you had more Legos, you could build this strand longer. This is showing 11 base pairs here, but I did that just to make sure that it would go around once, and if you wanted to make it longer, you could certainly add more base pairs on top. I also want you to see that I added some extra phosphate groups to the 5' end of each strand because we would expect to find triphosphate there. Anyway, I hope you enjoyed watching, and if you think you might like to learn how to build this model, keep your eyes open for another video I'm putting up which will take you through that process step by step. Thank you very much for your time.